Let's have a discussion on cell cycle in today's class. You know, all cells have to divide, like some cells like neurons. These cells have to divide for the continuous multiplication and the growth of the organism. You know, the cell cycle has been divided into many phases. M phase, then interphase. This interphase is again divided into three phases: G1 phase, S phase, and G2 phase. The M stands for the multiplication. G1, G stands for the gap 1 phase, then S stands for the synthesis or synthetic phase, then G2 stands for the gap 2 phase. So let's see what are the processes happening in the interphase first, because interphase is a preparatory phase for the cell division, and the cell division is occurring in the M phase. So in the case of G1 phase, protein synthesis is the first one, or the important step taking place, because the proteins, because all enzymes are proteins, these enzymes are needed for the DNA synthesis that is to occur in the S phase. And if there is protein synthesis, definitely there will be some RNA synthesis, because some RNAs are needed for the protein synthesis. RNAs like rRNAs, tRNAs, and mRNAs. And after that, for the necessary synthesis of DNA that is to occur in the S phase, there is need of ATP. That is also occurring in the G1 phase. ATP synthesis. And other raw materials, some minor raw materials are also taking place, synthesis taking place in the G1 phase. Then the total cell will grow in size. Raw material and the cell growth. These are the total process or overall steps taking place in the G1 phase, that is the gap 1. Then we can go to the S phase, that is very important phase, because if a cell is divided, the total genetic material has to be divided into two. The genetic material division is called DNA replication, because the DNA is the genetic material. The S phase, the S stands for the synthesis. The S stands for the synthesis, which is taking place in the S phase. So first one is the DNA replication. For the DNA replication to occur, there is a need of centriole division. Centrioles are necessary tubulin protein arrangements which are to be divided for the tracking of the DNAs to different parts or two parts during the cell divide. Centriole division. The next important protein, that is histone protein. Histone proteins are very essential for the packing of the DNA after the division, or after the DNA replication. They have to be packed and separated to different cells. So the packing proteins are called histone proteins. Histone proteins are synthesized in the S phase. But in lower eukaryotes, 50% of the histone proteins are synthesized in the G1 phase itself and 50% in the S phase. But in higher eukaryotes, total histone proteins are synthesized in the S phase. Now we can move on to the G2 phase. What are the processes taking place in the G2 phase? First one is the protein synthesis which are essential, or the proteins which are essential for the total cell division will go on or continue in the protein or G2 phase. Then RNA synthesis. As we discussed in the G1 phase, if there is protein synthesis, there will be RNA synthesis, because RNAs are essential for the protein synthesis. RNAs like rRNAs, mRNAs, and tRNAs. Then ATP synthesis will also take place in the G2 phase. These are the important processes happening in the interphase, G1 phase, S phase, and G2 phase. Now the cell is ready to divide, means division is called M phase or mitotic phase, in which there is important two steps, that is karyokinesis and cytokinesis. Karyo refers to the nucleus, so karyokinesis refers to the division of the nucleus into two. Means division of nucleus means total replication of the DNA, and replicated DNA will be separated into two cells. Then cytokinesis means total cytoplasm of the cell will be divided and the total division will occur. Then during cytokinesis, we can simply classify them according to the arrangement of the DNA, that is prophase, metaphase, anaphase, and telophase. These are the important steps taking place in the M phase or the karyokinesis prophase. During the prophase, the total DNA will be arranged or packed. Then during metaphase, the DNAs will be arranged in a particular plane called equatorial plate or metaphase plate. Then anaphase, the divided DNAs will be equally separated into two halves. Then telophase, they will be enclosed in nuclear membrane. The last step is the cytokinesis. During cytokinesis, the separated nucleus will be divided along with separate mass of cytoplasm. Then cytoplasm will be divided into two, that is cytokinesis, and the total cell division is over. Now that is the overall discussion, or the brief discussion of the cell division, and we can have which are the important steps happening in the prophase, metaphase, anaphase, and telophase in detail in the next class.